Morning everybody. Today I'll be presenting our technology, which is entitled Chia Seed Derived Products and the Process thereof. In terms of market need, it is estimated that the world population will reach 10 billion by the year 2050, and to grant food security, new sustainable food sources will play a major role in our diet in order to meet this global demand. From this aspect, it's known that chia seeds are highly nutritious, with a high content of fiber, lipids, and protein. However, the extraction of these compounds is often blocked by the presence of a highly viscous fiber called mucilage. Our technology mainly focuses in the efficient separation of mucilage using a green approach. This not only increases the yield of extraction of mucilage, but other components as well, such as proteins and fats. This technology also enhances specific attributes which make the products suitable for commercial applications in pharmaceutical, cosmetical, and food industries. Some industries that might be interested in our technology are Cargill, ADM, Mead Johnson, The Ordinary, L'Oreal, and Olay. In the case of the market need, it is estimated that the global protein ingredient market share was 38 billion in 2019. Our technology mainly focuses in two aspects of it: the food and beverage industries and the personal care and cosmetic application industries. And in this aspect, the plant protein market is expected to expand from 10.3 billion to 14.5 billion by the year 2025. Therefore, innovations and technologies focusing in the extraction of plant-based protein and components will be highly valuable in upcoming years. Finally, plant-based ingredients market size is projected to increase to 13 trillion by the year 2025. In the case of our technology, we apply the combination of ultrasonication, temperature, and vacuum filtration to efficiently separate the mucilage component from chia seeds. In this aspect, this yields a highly pure fiber, and it also enhances the extraction yields of proteins and fats. After the fats are extracted by cold oil presses, we also incorporate the technology of proteolysis. This produces peptides that are essentially small fractions of proteins that have enhanced functionality and activities. So in this aspect, the chia seed peptides have shown to have overall high antioxidant protection in cells, a high capacity to inhibit enzymes that are related to skin aging, a higher techno functionality in foods, and also they offer benefits to diabetic and hypertensive people. Our technology applying ultrasonication and proteolysis using commercial enzymes offers a high yield of protein extraction and high quality byproducts as well. This technology has shown to be superior to other technologies out there, such as convectional heating, which involves drying the fiber component and using sieves to separate it, or using freeze drying techniques and sieves to physically separate this fiber component. This technology is faster, offers a high purity, and more importantly, it enhances the biological activities of these components for pharmaceutical and food applications. As for the next steps, we would like to partner and collaborate in the future with companies such as ADM, Cargill, The Ordinary, and Olay. This in order to develop food products or cosmetical products with chia seed components. We will also like to explore primary investors that might be interested in the commercialization of chia seed products and byproducts. Thank you so much for your attention, and I'll be happy to answer any question. Have a nice day.
Hello, everyone. I'm Rudy Banerjee. I'm here to present you Skyros, the last mile risk-free autonomous delivery system. Skyros actually is trying to solve a problem which the present industry is not being able to, which is that failure. And we have drones which are being used by, which are being developed by companies with the help of FAA, but FAA model is definitely going to fail. The problem is that we already have this promise, which we are hearing from Amazon, 2-hour delivery, and then Amazon is saying you can call Alexa and say, tell KFC I'm hungry. So shopping lists are going to go away. We already make 500 billion trips every year to the store, to errands. And it is going to increase if we don't have shopping lists, or if we have smaller refrigerations and all in our house. We are going to just order without looking at it. So from 500 billion, it will become 2 trillion in the next decade or so if we have instant delivery and autonomous deliveries and within the price points. One problem is that a lot of people are not going to accept it in the beginning, and they have a lot of power. So city, unlike FAA, cities, local governments, they have a lot of control over their over the landscape, and so they're going to ban drones if they kill pets or babies or anyone, one thing. And that happened in Arizona. Actually, there was one death of an effort by Uber, spent $1 billion to develop autonomous vehicles on the road, and there was one death, and they had to stall that $1,000 million investment. So I don't see drone companies being successful there, but I have a solution to it. Why don't we hack the sky in such a way like we hack the underground, like railway tracks, the subways, and we hack the sky. We give rights to the city and the companies, limited number of companies, to fly drones and make it more legitimate, and also have local security police control that space. So I'm going to talk about just the basic market need. As I said, it's already a $2 trillion last mile, but it's going to reduce it a lot because of automation or autonomous vehicles and all. But we still have a big market to do, because with no shopping lists and all, it's going to increase. But even without that increase, even with a very small percentage, there'll be $10 billion for us to work with in the beginning. So what is the technology I'm proposing? Unlike straight line geometric networks used by FAA to fly 1 million planes a year, for 1 trillion drones, we need something like what we have on the ground, which is topologically designed roadway systems with right of ways and other things, and traffic furnitures and other things like stop signs. We don't need that sophisticated ground level stuff, because in the sky you don't have pedestrians. But if we actually specify a very small, tiny percentage of the airspace through a regulated airspace of sky routes, then which replicate the ground level, which is also public space, and the cities actually have control. And if the FAA actually, instead of the FAA, the local cops control the transportation, then we are going to have really successful outcome. So if you see in this picture, actually Skydos shows those drone being delivered just like you would be on the ground. The technology space actually up there in the sky, actually what is happening with the companies like Amazon and all is they're trying to perfect the airspace by having a better and better drones. I say that even with poor quality drones, if you fly it in structured airspace, it'll be a solution. So it's a topological solution through a structured airspace, not the unstructured airspace, which is used by more than 100 companies now. And that is the FAA model. My model is, I call it the FCC model. We'll auction that a small airspace, very tiny airspace at 300 to 400 feet, or at least just one level, like 300 feet in the sky, which would be under the control of the city. The next steps, I actually need a lot of help from people who are developers, like drone engineers and app developers, and also I need investors. I need to do a proof of concept, which is basically a very small place like a campus or a university or maybe a city, and let people just order things. And for that, I just need generic drones, and I actually hack the sky with the software, and we need some end software that would be in use. Thank you very much.
In 2008, a sugar refinery exploded in Fort Wentworth, Georgia, that killed 14 people and injured about 40 workers. Unimaginably, the reason for explosion was dust. In this presentation, I'm going to talk about my technology on preventing dust explosions through real-time measurements. With most of the food, grain, pharma, and chemical products are handled in powder form or processed in powder form, these operations generate lots of dust. You may have seen these published reports. In 2019 alone, there were 75 reported explosion incidents that occurred throughout the world. Other than fatalities and injuries, these explosions most times destroy the whole facility. So when started, this facility, as shown in this picture here, was the largest grain handling facility in the world. But as I mentioned before, in any grain handling or any kind of powder processing industry, dust is omnipresent. In all these facilities, these 5 factors like dust, dispersion, ignition, etc., are present always during the operation. With all these 5 factors, the powder processing operations will be a ticking time bomb. And when we have all the 5 factors present, it can lead to an explosion. So in this explosion that happened in the largest grain handling facility in the world, 7 people were killed and about 10 were injured. And you may know that OSHA is the authority that develops regulations for the industry to follow. So if OSHA authorities visit a facility and observe dust in the premises, the minimum fine will be close to about $13,000. But if an explosion occurs, the financial loss could be multiple of millions of dollars. For example, if a minor explosion occurs in a facility whose revenue is more than $50 million per year, a 3-day of downtime can lead to financial loss of about $400,000. And this does not include OSHA or local authority fines, increased insurance, injury or fatality compensations, rebuilding costs, lawsuits, etc. So how can we prevent these deadly accidents and the potential financial loss? For the dust to explode, the concentration level should reach a minimum explosive concentration. With my experience of training industry workers and based on my research, my team came up with this technology of measuring suspended dust concentration. In this technology, we use a 2-target method to determine the suspended dust using light extinction. With no technology to measure suspended dust concentration, this technology will be a much needed intervention that the industry needs. And this technology has been calibrated to determine dust levels of above 15 grams per meter cube. Our major competitors are dust collectors or dust monitors, but these instruments can help collect dust and then determine the concentration. So there is no real-time measurement of dust concentration. The explosion suppression devices or venting devices can help reduce the magnitude of an aftermath of an explosion. So they are not warning systems that could help mitigate an explosion before it occurs. So our technology can be integrated with process control software for continuous monitoring and will be a tool for continuous measurement of dust in the facility. Another major advantage of this technology will be that this does not require any training for the people to use. In summary, to prevent any dust explosion from occurring, the industry needs a forewarning technology that can alert industry workers about the formation of an explosive atmosphere. Our technology can continuously monitor the amount of dust within the facility and also allows for remote access. To further this innovative concept, I am looking for partnership to take this technology to the market. And thank you very much for listening to this talk.
Hi, I'm Dr. Kara Stewart, a member of the team of scientists of Fertilipid, also including Dr. Teresa Casey and Dr. Christina Ferreria. In the US swine industry, we kind of have two populations of animals, those that are going to be raised to become meat for human consumption, and those that are raised to become the breeding females to continue producing pigs. Today, we're going to focus mainly on just those breeding females. These young females are selected at birth and only have a 53% chance of becoming a breeding female. And this is because they are typically removed for poor fertility. As of today, we have 6.3 million breeding females in the United States, and they have a 50% annual replacement rate. That means we need 3.15 million young females every year to be produced to replace those females that are being culled. That means we need to raise twice as many as are needed because they are being removed at a rate of 53% for low fertility. So if we look at the table here, we can see what the costs are associated with raising these females that will never actually enter into the breeding population. So every time you raise one breeding female up to the age of puberty, it costs the farmer about $172. If she ends up being infertile, then she will be sold in the meat market for human consumption, and her value is about $165. So this results in a net loss of $7 per animal raised that does not end up contributing to the breeding population. Collectively, this is a $22.5 million loss per year to the swine industry to raise these females that will end up with poor fertility. So what if we could identify these infertile females at a young age and only raise the ones that we know will be fertile? Well, Fertilipid has identified infertile females that have a different lipid composition in their vagina at 3 weeks of age compared to the fertile females. So our technology uses a small brush to enter into the piglet's vagina and obtain a sample of cells from the vagina. Those swab brushes can then be sent to Purdue, where we use mass spectrometry using a rapid detection method that was invented here at Purdue to determine the lipid profile of the cells of that pig's vagina. Based on the lipid profile, it's not just one lipid, it's a series of different lipids that are altered in these infertile animals. We can send results back to the farmer to identify females with the lipid profile associated with infertility so that they can remove them and not select them at weaning, which is at 3 weeks of age. This technology is truly novel, and there is no other technology like it on the market today. And what we envision for this technology is similar to like a genetic test that you would take at home, like a 23andMe, where you would receive the Fertilipid kit on the farm, you would swab multiple pigs and label the tubes with the brushes, send them to Purdue, and we would report back the results to the farmer. And that way the farmer can save millions of dollars a year, a true economic savings by being able to better select fertile females to continue to raise to puberty. So currently we have really great data that confirms this lipid profile in only one large commercial population of animals. So we are looking for investors and industry partners that can allow us to gather some data in additional animal populations to make sure that we have the full lipid profile identified. We'd also like to investigate whether buccal cells, which are the cells inside the cheeks, could be used in addition to vaginal cells in order to have maybe a more easy collection method. Then we would like to perform some customer interviews and create our business model, and then create the prototype and validate the sampling kit to send out. So Fertilipid is really excited about our new technology, and we look forward to having discussions with you.
Handheld device for biodefense and food analysis. Foodborne illness costs about $78 billion per year in the USA. The Centers for Disease Control estimates that 48 million people become ill, 128,000 are hospitalized, and 3,000 die of foodborne diseases every year in the USA. The marketplace needs rapid detection tools for biodefense and food analysis. Food fraud is rampant worldwide. This is the practice of adulterating food for economically motivated reasons. The impact on the food industry is in excess of $50 billion annually. Contamination of food can happen in any step of the production process from farm to table. Rapid technologies are needed to solve this serious issue. New applications for food authentication are excellent commercial opportunities. We are focusing our efforts on the most impactful applications such as harmful microbes, toxins, and chemicals. Detection of allergens, pesticides, and analysis of food processing equipment and raw food materials are also potential targets. We are not limited to food. There is a huge need for effective detection of biowarfare agents during military surveillance, as well as biodetection for environmental monitoring. According to Frost & Sullivan reports, the market size for food safety testing is large. It is projected to experience more than 7.5% CAGR from 2019 to 2025 to reach over $24 billion. This is not just a US market, it is global. The food authentication testing market is also significant. This is particularly true for higher products, which are more likely to be forged with lower quality or entirely fake materials. Driven by end-user requirements, there is an increase in demand for portable instruments to perform simple chemical analysis for field-based applications. Based on the promising market growth predictions and significant market revenue, we see our handheld technology as being very profitable, given the significant market need. Our group is developing a handheld laser-induced breakdown spectroscopy instrument and assay technology to detect food contaminants and identify food authenticity. It is real-time and multiplexed, because that is what the market is now demanding. This handheld LIBS technology can be used to analyze the food samples for authenticity without significant sample preparation. Machine learning algorithms are directly incorporated in the handheld device. This will deliver a simple and fast identification and classification procedure for quality control of food samples. To summarize the biodetection procedure in a few steps, it includes liquefying the sample, applying it to a bioassay substrate, analyzing it with atomic spectroscopy, and a clear readout of results. The device is portable, allowing food inspectors to go from one facility to another, testing products and geotagging their locations. It is multiplexed, allowing the users to detect multiple contaminants in one single hit. Our first U.S. patent of the bioassay was issued on December 24, 2019, and another related full patent was submitted on September 17, 2019. A PCT is under review. These patents cover the principle and many potential uses of this powerful technology, which is capable of identifying all the elements in a material in one single sampling event. These patents secure our technology in the legal landscape. The figure on the right demonstrates that feature selection algorithms can effectively classify and identify a variety of food samples. So what defines effective contaminant detection? Our technology combines sensitivity, specificity, and ease of use. But it also shows the ability to detect microbes and chemicals while multiplexing at speed. While PCR and immunological detection are important, none of these technologies share a combination of all the key features of our handheld LIBS technology. Currently, Thermo Fisher and Agilent are two companies that dominate the food analytical device market, but their LIBS devices can only detect metals. No one has a handheld device for biodetection. Thus, there is significant space for our technology. Our team is diligently working on the development of our handheld LIBS device and the associated assay technologies. We are interested in collaboration with potential manufacturing partners and investors who would like to help bring this technology to market. We look forward to discussing more with anyone interested in this forward-leaning technology.
Hello, my name is Mohit Verma. I'm the founder of Krishi LLC. I'm also an assistant professor at Purdue University. Today, I will talk to you about Discover, which is a platform of sensors for detecting bovine respiratory disease. Bovine respiratory disease affects almost 20% of the cattle in the United States. Currently, in order to diagnose bovine respiratory disease, the farmer has to walk around and look for symptoms. For example, changes in breathing, loss of appetite, depression, as well as elevated temperature. The treatment of bovine respiratory disease is based on trial and error. So you pull the cattle aside and give them antibiotics. This treatment fails 1 out of 3 times, and that failure leads to losses of almost $1 billion per year in the United States. The failure happens because the farmers and the veterinarians don't really know which pathogen is causing the disease. We are trying to build a sensor that can tell you which pathogen is causing the disease and therefore improve the treatment efficacy. We're targeting the market of animal diagnostics, starting with cattle diagnostics in the US, which is in the order of $29 million, and using a platform technology to also target other animals, which brings us to the animal diagnostics market in the US of about 1.7 billion and global animal diagnostics of almost $3.1 billion. To target this market, we introduced Discover, which stands for Diagnostic Sensors for the Cause of Bovine Respiratory Disease. These sensors work as follows. As you see on the right, you would collect a nasal swab from the cattle that you think is sick, and then you would put this nasal swab in our device, which is shown in the blue box. The device will heat up the sample and conduct a reaction to detect the pathogen. After about an hour, the response would be sent to your smartphone in terms of which pathogen is present and also what is the most likely treatment. Using this approach, we can deliver animal diagnostics on the farm, provide results in less than an hour, improve animal welfare, increase the judicious use of antibiotics, as well as eventually increase the profit for the meat. The technology is based on 2 parts. One is the cartridge, which is these paper-based sensors shown on the left, and another is a heater imager device shown on the right. The cartridge is where all the reactions take place. This is where you can detect different types of DNA or RNA from the pathogen and cause a change in color. The different pathogens are shown in those red circles in the device. The cartridge goes into the heater, which conducts a reaction and also captures images so that you can see the color change. And the heater can be operated by a touchscreen that's present on the front and also using your smartphone. With this technology, we are competing against existing diagnostics and therapeutics. From the diagnostics perspective, lab-based diagnostics can be quite slow, as we've seen with the COVID-19 pandemic, for example. Portable diagnostics, which are being developed, only use symptoms, don't really tell you which pathogen is present. And symptoms-based diagnostics is limited because, again, it doesn't tell you which pathogen is present. From the therapeutics perspective, you could use vaccination, but it might not be effective if the pathogen causing the infection was not included in the vaccine. And you could use antibiotics, which is the current standard, but it also fails 33% of the time. So Discover comes in by making a user-friendly test that can be used on the farm and by increasing the efficacy of the therapeutics to a target of 90%. Right now, we're looking for business leaders to partner with so that we can bring our proof of concept to a product. We're also looking for expertise in the animal diagnostics market so that we can enter the market and help avoid pitfalls and improve product features. Thank you all for listening.